So mitochondria, ribosomes, and lysosomes are considered to be, goes without saying, they are organelles of the cell. Do we need to go through what they do and maybe others?
Just that they are building blocks. I don't know if there's anything to add, like cell is the basic unit, then tissues are made up of cells and organelles are made up of different.
Yes. Yeah, and then maybe we can say mitochondria, we know it's the powerhouse of the cell. Ribosomes, protein synthesis, and they're present in the rough endoplasmic reticulum and the cytosol as well. We have the, what else? Lysosomes. Lysosomes, they degrade excess and worn out parts of the cells. Yeah, they also break down the macromolecules. We also have the endoplasmic reticulum, smooth and rough. Rough has ribosomes, smooth has nothing. And smooth usually deactivates drugs. It's responsible for deactivating stroke inactivating drugs. What else? Any other, apart from the centriole where cell division occurs? And the proteasome, which breaks down denatured and damaged proteins. Is there any other that I'm forgetting? The cell organelles. So that goes without saying that the next question, the answer will be the mitochondria, because we just said it's the powerhouse of the cell. Then number 47, all the following are APCs for helper T lymphocytes except its neutrophils, because dendritic cells, I remember we once said they're the professional ones. We also have macrophages and B lymphocytes. So it goes without saying. Yeah. Then 48, 46 is A. Because, yeah. 48, the most immunogenic molecules. What is the meaning of immunogenic? I'm thinking it means immune genesis. So I don't know if that's true, but
immunogenic means something which is capable of causing an immune reaction.
yes. So immunogenesis. And we know most of them are usually proteins. So it goes without saying that they're proteins. Most of the immunogenic toxins, blah, 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 proteins. Most of them are usually proteins. (49) Dash is the most abundant immunoglobulin. We said it's IgG, and the least is IgE. So it's IgG. Then, is there anything that has not been discussed under immunoglobulins? I don't think so. I think that was covered sufficiently. Then innate immune system consists of, so maybe let me start from choice D, complement system and IgE, IgG and IgD, B and T lymphocytes. We know these are part of the acquired. So the neutrophils and macrophages are part of the innate. Moving on swiftly. Yeah, so the innate system, it has
Diana,
the
sorry, I have a question. You
Mhm.
said that the list is IgE, and I was wondering, isn't it IgD?
We said IgE just juicy, just the
Okay.
other day. So I don't know. Maybe we look at the percentages. Maybe let me confirm.
IgE is like 0.002% and IgD is 0.2%. So IgE is yet the answer.
Oh, it's the list, yes.
Okay.
Yeah.
So we were talking about the innate. We said the innate, it's non-specific. So it's composed of the natural killer cytotoxic cells, complement, neutrophils. So complement is part of the innate. Then for the adaptive, which is specific and it has memory, we have the cell-mediated and antibody-mediated. So cell-mediated has the helper T cells and antibody-mediated has the B cells. So complement is part of innate as well as neutrophils and macrophages. So the answer is A. Has Janet showed up?
Diana,
Hmm?
you can continue. I'm a Stanley.
I cannot.
Stan will help you.
Okay, maybe he can continue from 9, maybe. So cytokines. So cytokines do not participate in immunological responses. That's false. They are responsible for induction of erythropoiesis. That's not true. We know erythropoietin is the one responsible for that. They are produced by T cells and they occur in only one form. So what do you guys think?
By elimination, C.
Good. It's yes, they are produced by T cells. we don't want to go through, or you guys want us to talk about how cytokines are produced, but I don't think so, since the question will be MCQs, so I don't think that's really necessary. 52, the mediator of type 1 hypersensitivity reactions is, we know it's IgE. We've gone through this a couple of times, so maybe a recap. IgE is type 1, type 2, and 3 is IgG, then Ig, then type 4 is T lymphocytes. Helper T. Then which is an example of type 3 hypersensitivity? So allergic rhinitis is part of, is it type 1? Is it type 4?
Type 1.
Type 1. Lepromatous leprosy.
Type 4.
And finally, asthma. Is asthma type 1, type 4?
Type
Is
1.
it caused by an allergy or? But we need to know that chronic asthma is type 4. Chronic asthma is type 4.
Allergic asthma is type 1.
Yes. So we know glomerulonephritis is type 3, together with rheumatoid arthritis, serum sickness. I remember of serum sickness, and RA and SLE, the part of type 3. So number 53 is B. '54, natural killer cells are found only in adults. That's false. Are a subset of macrophages. That's false. Phagocytize infected cells. Do they do that? Do they do phagocytosis?
No, the cytokines, they produce some chemicals through cytokines.
So the answer is A. Moving on
I
swiftly.
don't know if we've talked about natural killer cells or if we need to talk about them. I don't know.
Okay, we can talk about that. Okay, so I was getting some reliable notes on natural killer cells. We know they're part of the innate immune system. and they produce gamma interferon. So I know that choice that we've picked, maybe we're wondering what's that? That's gamma interferon. So they're responsible for that. So the natural killer cells, they don't phagocytose, they activate macrophages. Now the macrophages are the ones who are responsible for killing the bacteria. They act similarly to cytotoxic T lymphocytes by participating in apoptosis and all that. Yeah, so I think that's what is really important.
From which stem cell is it produced? Is it the myeloid or the lymphoid?
I'm thinking myeloid because lymphoids, we know it's for B and T lymphocytes. Or
I was
it could be
confused. I thought
lymphoid.
it's from lymph. I thought it's from
Pardon?
lymphoid because
Oh,
I
wait,
thought it's
it
from
could
lymph.
be.
I'm not
Yeah,
sure.
yeah. Because here it's saying approximately 5 to 10% of peripheral lymphocytes are natural killer cells. So they could be from they're actually natural killer cells are lymphocytes. So it's from the lymphoid. But for them, they don't pass through the thymus. We know the T lymphocytes, they pass through the thymus in order to mature. But for natural killer cells, they don't. They don't have to pass through the thymus to mature. Yeah. So it's the lymphoid. Sorry for that. Then antibodies. Natural killer cells can increase red blood cells and reduce the need for in patients receiving chemo. Okay, if that could be true, let me see. Maybe is that true? Can we say that's what natural killer cells do in patients receiving chemotherapy, according to the chart?
I think it's true because they are developed in the bone marrow as well as extramedullary sites such as lymph nodes, thymus, liver, and uterus. So if they're developed in the bone marrow, it is likely that they increase the red blood cells. If that makes sense.
Natural killer cells. Okay, I'm seeing the functions of natural killer cells from an immunology textbook. They just kill virus infected and cancer cells. And their killing is non-specific and not dependent on foreign antigen presentation by class 1 or 2 MHC proteins, and they produce gamma interferon that activates macrophages to kill ingested bacteria. So I think the most appropriate answer is secrete gamma interferon. About the about erythropoiesis, personally, I'm not sure. But there might be a role, of course. Anything is possible with science, though. So, oh, it's true. Okay, it's true. Antibodies are produced by B lymphocytes. passive immunity is since they're developed in the bone marrow. Okay, so we can say natural killer cells promote red blood cell production. Yeah, maybe. Yeah, so maybe we can give our contributions on passive immunity before we answer the question, what do we know about passive immunity? What is the other name for passive immunity and the likes? Okay, maybe I can. So the difference between active and passive immunity. So the mediators of active immunity are antibodies and T cells for active, but for passive it's antibodies only. Okay. Then advantages of having active immunity, just from the word, it stays for a long period of time, so long duration. And for passive immunity, it's immediate availability. That's the advantage of passive immunity. It's immediately available. Then, since it's immediately available, the disadvantage of having passive immunity is it's a short duration. So just a period of like months. And the disadvantage of active is that it has a slow onset. It takes time before it kicks in. So an example of passive immunity includes administration of antibodies against maybe tetanus and all. Those tetanus, diphtheria, So when you administer the antibodies, against those organisms, the body makes large amounts of antitoxin immediately to neutralize those toxins. So that's what happens. Another form of passive immunity is when IgG is passed from mother to fetus during pregnancy and IgA passed from mother to newborn during breastfeeding. So that's the other form of passive immunity. So according to the choices, our answer will be B because we've said it's short-lived. It's available just for months, just for a few months. Any other additions to that? Just like Dr. Tour has sent, acquired immunity, it's either active or passive. So active is you develop after being exposed to an infection or from getting a vaccine. So it's natural or artificial. Artificial is from vaccination, natural is from exposure to an infection. But for passive, you acquire from someone or something else. Someone includes mother to baby, either during breastfeeding or during pregnancy, where we said IgG is during pregnancy, IgA is during breastfeeding. And artificial antibodies are acquired from an immune serum medicine. But passive is short-lived, active stays for a long period of time. Then the gland that secretes cortisol it's the adrenal gland. Yeah. It's D. Maybe we can give our contributions on its secretion, like what triggers its secretion and all. Someone please. Someone can help us trace how cortisol is secreted. Shafi. Yo. Okay, maybe we just go to the next question. So, which of the following glands needs iodine to function correctly? So we know it's the thyroid gland. Yep. Then the universal donor is a person with blood group. Oh. So we know when a person has blood group B, they have B antigen and A antibodies. So it means they can only receive blood from someone who has type B or someone who lacks the antibodies against type B. Then when a person has type A, they have A antigen but B antibodies. So they can only receive blood from a fellow A or someone who doesn't have antibodies against A. Then AB, they have the A and B antigen, but they lack antibodies. So that's why they are usually the universal recipient. But O is a universal donor because O has no antigen as compared to the others. So it's O.
Please repeat again.
Sorry. So I was saying when a person has, so we know the surface of the red blood cells, of everyone's red blood cells in this meeting has an antigen. Personally, I am blood group type O. So I am a universal donor because my red blood cells, they don't have an antigen. So that makes it easy for me to donate blood. But I have antibodies against type A and B. So I can't receive blood from a type A or B. I can only receive from a fellow type O. Type people with blood group type A and B, they have A and B antigens on their red blood cells, but they have no antibodies in blood against, So that's why they're the universal recipient, because they have no antibodies. But type B has a B antigen on the red blood cell, but A antibodies. Like it's opposite. If you have a B antigen, you have A antibodies. You have an A antigen, it means your blood type is A, so you have B antibodies. Is that in order or still confusing? So if you have
Thank you.
type, oh, sawa. So it's A, universal donor. So the hemolytic disease of the newborn is it erythroblastosis fetalis? Is it hemophilia? Is it leukemia or anemia? We know anemia is what is anemia? Maybe let me know. For contribution. Man, you people should contribute at least kidogo. Even in the chats, basi. Yeah, we know the answer is A, but maybe there's someone here who doesn't know what hemophilia is, leukemia, anemia.
Anemia is a condition in which the body does not have enough healthy red blood cells.
Leukemia, we know it's white blood cells, and hemophilia.
So we said, I think we said hemophilia, there's hemophilia A and hemophilia B. So A, like factor, is it 8?
A.
Yeah. Hemophilia B, you lack 9. Ideally,
Mhm.
hemophilia when you have uncontrollable bleeding. Yeah.
Good. So good. So it's erythroblastosis fetalis. That's the term. Sinoatrial node. Is it located in the left ventricle? No. Does it beat faster than the rest of the myocardium? Is it synonymous with the atrioventricular node? Does the sinoatrial node not influence the blood flow? So by elimination method, we know D is out.
C
So
is
the
out.
answer is, D is out, A is out. Is it synonymous with the atrioventricular node? No. So it beats faster than the rest of the myocardium. And I think was it in today's session, one of the sessions, they talked about how it flows from the sinoatrial node to the atrioventricular to everywhere up to the Purkinje and all. It's in the group. The photo was sent in the group. So diabetes insipidus, I think this has been talked about a couple of times, and it occurs due to lack of vasopressin or ADH. So that's B. Then glycogenolysis. From the word genolysis, it means lysis
I
of.
have a question.
Mhm.
What is the drug of choice?
For
The diabetes insipidus.
okay, maybe we can discuss that. I think a photo was sent. A photo was sent in the group, summarizing all that. Maybe we can discuss that.
I think you can give vasopressin.
Yes. Something else. Yes, desmopressin. That's the first line. There's also hydrochlorothiazide, amiloride, indomethacin. So since we know thiazide diuretics, maybe I confirm. I think first line treatment is sufficient, plus the diuretics. The thiazide. Mhm.
That's okay. Thank you.
No.
So can I read about the diagnosis?
You can.
Yeah, the test is water deprivation test. For this
Mhm.
test, you stop drinking fluids for several hours. During the test, your healthcare provider measures changes in your body weight, how much urine your body makes, and the concentration of the urine and blood. Then, also, they measure the amount of ADH in your blood.
Okay. Thank you. Any other addition? None.
I don't know, but if we've talked the different types of insipidus, diabetic insipidus, have we?
Yeah, we did. Gaius covered that, I remember. Or did he?
Yes, we did.
Yeah, I remember. I
So
remember he
for the
covered
treat-
that. Mm-hmm.
for the treatment is different for the different types. For central diabetes insipidus, I don't know if this was mentioned before I joined, but for central diabetes insipidus, that means we're having insufficiency of ADH, right? So that's when we give desmopressin. We give desmopressin, yeah. And then for nephrogenic, it's that the kidneys cannot respond. So that's when we use thiazide diuretics, amiloride, endomethacin, yeah.
Mm-hmm.
Thiazide
The end.
diuretics only. That's the one we had mentioned, I think.
Yeah.
Yeah, for the mechanism of action of how they work, I think it's an opposite, whatever. Like, it doesn't make sense,
Yeah.
but that they use it against it.
That's true.
The types you said it is
Really?
central.
Is central, there's nephrogenic.
Okay,
Yeah.
thank you.
Mm-hmm.
What
So
about
what is
gestational?
what? Pardon?
What about the gestational diabetes insipidus? How do you treat that?
I think Gas had said, if I'm not wrong, you give, yeah, cause it may lead to the, like, so we expect them to, eventually affect gestation.
Pardon.
So desmopressin is used for central and gestational. Then nephrogenic is for thiazide diuretics, which will reduce the amount of urine, of course, your kidneys produce. And the NSAIDs. Is that in order?
Thank you.
Okay. So maybe I can cover until number 65, then stano and continue. So glycogenolysis, from the word, it's the lysis of glycogen. It happens when there's low glucose levels in the body. So should we go through the cycle, or is it really necessary?
You can remind us.
Okay, I can do that. So, this enzyme, it's called glycogen phosphorylase. It's the one that's responsible for breaking down glycogen to glucose 1 phosphate plus glycogen. Now we know that glucose 1 phosphate will be converted to glucose 6 phosphate, and now it will get into the glycolysis pathway. So how does glycogenolysis, like how is it initiated? It's initiated by, sorry, by glucagon
Yes.
Yes. And in the liver, glucagon will initiate it. And in the muscles, it will be the epinephrine. Epinephrine will stimulate glycogenolysis in the muscle and glucagon in the liver. So,
About cortisol.
But I think it also does the same.
Yes.
It has yeah. So I've said that because I know glycogen is stored both in the liver and the muscle. The liver to maintain blood glucose levels and the muscles to meet energy requirements. So there's that. So maybe someone was disputed thinking, I think glycogenolysis could be taking place only in the liver. No, doesn't occur in the liver only, it also occurs in the muscles. Does it occur in the kidney? No. And is it stimulated by hyperglycemia? No, it's stimulated by hypo. So the answer is automatically B, since it's initiated by glucagon but inhibited by insulin. All the following are functions of Sertoli cells except, I think we covered this, I think in night session. Is that correct? I think we did. So I remember we said Sertoli cells are responsible for production of the androgen binding protein, whereas Leydig cells are responsible for production of testosterone. So I think my device is going off. Maybe is it Stano or Janet? Can continue from there. Janet, please, Mystic.
And we were having the discussion. Anyway, so
Please, thank you.
You finish with 64? You say the answer is? Diana. Okay, so basically she's disappeared. So apparently this question was discussed, I don't know when.
No, we just they produce that androgen thing. We didn't discuss the functions of the cells.
Oh, we didn't discuss. So it is used in sperm production. I don't know if it phagocytizes defective sperms. Nourishing. Okay, what are the functions of the Sertoli cells? Let's see. Do Sertoli cells phagocytize the defective sperms?
Sertoli cells are the somatic cells of the testes. Sertoli cells are the somatic cells of the testes that are essential for testis formation and spermatogenesis. Sertoli cells, they facilitate the progression of germ cells to spermatozoa via direct contact, and by controlling the environment, may live within the seminiferous tubules. Formation and spermatogenesis.
So by elimination, even by elimination, I don't know. Do they secrete inhibin? Because production, except production of sperms. I thought you just said they produce sperms. They're using the production of sperms. So that can't be the answer.
Okay, wait, I read another one. One, there are 4 functions. 1, supportive and trophic functions for the cells of the somniferous epithelium. 2, transport of mature spermatids towards the lumen of seminiferous tubules. 3, secretion of androgen binding proteins. 4, production of substances with endocrine or paracrine action.
So they do everything from A to D.
I don't think they produce sperms.
They don't produce sperms.
No, they just nourish and develop them. I think that's a different thing.
Okay.
Maybe.
Production of sperms is by the testes, not the Sertoli.
Okay. Okay. I think they're essential for spermatogenesis. All right. The next one. So that one was, it doesn't produce, you said it doesn't produce sperms.
Yeah, that somebody has written something on the chat. Maybe you can read
On the
it
chat?
out. Yeah.
Oh, so so somebody says Sertoli cells are not responsible for producing sperms. They are a type of tentacular or nerve cell found in the human testes, which contributes to the process of spermatogenesis. Is a structural component of the seminiferous tubules, so they only contribute, they don't do the work. They provide nutrition and shape to spermatozoa and develop germ cells, secrete a number of substances that maintain and stimulate spermatogenesis. They produce testicular fluid, including a protein that binds and concentrates testosterone, which is essential for the development of the protozoa, and help to translocate the differential cells to the lumen. This is the one which was being read by, somebody was reading this. We already been read for this one. But okay, so they do not produce, the sperms. That doesn't include the production of HCl in the stomach. The acetylcholine, the gastrin, histamine, and the glucagon. I'll go with, let's see what you have qualifies D. I agree. That's a good D. Let's move on. Because I think the other choices are kind of there. All the following are plant growth regulators except auxins, gibberellin, kill cytokinins. Have we other than the time we were doing the discussions, have we like done like a crash on these hormones and different other questions? All right, let me look for a crash one. So some will tell us the function of the auxins or type it. The auxins, the gibberellins, the cytokines, that abscisic acid. Yeah?
I'm saying the auxins promote apical growth, so growth upward.
Apical growth, so growth upwards.
Yeah.
Okay. And the lateral growth is promoted by? I think the lateral is by the cytokinins.
Yeah.
Yeah, by the cytokinins. The gibberellins, what do they do? Okay. So ideally, the gibberellins are used to help in breaking. Something induces the seed dormancy, and I know gibberellins help in breaking the seed dormancy.
No, it's
I
the
don't.
abscisic acid that is for seed dormancy. Gibberellins are for stem elongation.
Yeah, gibberellins are for. I think abscisic acid will promote the seed dormancy, and then the gibberellins will do both breaking and also the elongation, the bud elongation. Amma?
Correct.
Thank you. And then we have the ethylene for fruit ripening. So which are the following plant growth regulators? All the following regulators except auxins, gibberellins, growth regulators. I don't know what that one is. What those are? What is salicylase? Let me tell. No. Since it looks like a whole family.
Yeah.
It's an order in a dichotomous plant with the Salicaceae family. Hi. All the following are some this one we did. Not we did this question though. Benzoyl, Wolfart, Macete, Canuba. I also go with benzoyl. I think we'd covered the waxes. Dash is for deoxysugars in fat. Dash test is for deoxysugars found in cardiac glycosides. Did we mention the test in the morning? No, we didn't go to the test when you're talking about the cardiac glycosides.
Janet, bongo. Ilituma kwa group.
Ilitumwa? Ilitumwa kwa group. Gani ilitumwa kwa group? 67 na 68.
Tests zote 61. Hii 68 sorry.
68. Hela Kilian, okay. I guess you can a group. That's for the two main groups of tannins. You do have a group letting.
What's it?
Tunaambia each other two main. Okay, amesema D. If I'm not wrong, if I had her right. Hydrolyzable and condensed. Is that what you said?
Yes.
Yeah, okay. Cleaning from the Syntona tree bar is a or an ethanol alkaloid.
Before we move on, can we do the tannins? Like somebody can do quickly an overview.
Oh, okay. Yeah, so somebody can give us an overview for the tannins. Kindly. Who is Stanley? Stanley, sing for us your song. Seems like we don't have much information about tannins.
Okay. So tannins are naturally occurring polyphenolic compounds and they can be found in various tissues. They are very different from others because of their astringent taste and the properties of tannins is of astringency. So it's just because of the examples is like unripe fruits, red wine, and black tea. And this property arises from their ability to precipitate proteins. Then chemical structure, we've said that they can be classified into hydrolyzable tannins and condensed tannins. So hydrolyzable tannins are esters of gallic acid and glucose. So they would be found in nuts, fruits, and subherbs. Then condensed tannins are also known as proanthocyanins, and they're made up of flavin-3-ols. So this is the reason why some of them are like, they have that red color. So example would be apples. And they exhibit antioxidant activity. The sources of tannins are bark, leaves, fruits, and nuts. Yes, that's it.
Thank you so much, Rina. So you can go on. That's the botanical source of ginger. Dr. True has told us it's Zinga officinale. Zingiba. I agree with her. Hi. All the following are used in aromatherapy except we have
On tanning, I wanted to add the test for tannins, ferric oxide test. Ferric test.
Ferric test.
Yeah.
Okay. Thank you.
What would be the color or the result of it?
Gray to black. A gray to black color formation indicates the presence of tannins.
So will they precipitate or just a color change?
I think it's just a color change.
Thank you.
Welcome.
Okay, so we are the plants used for aromatherapy. Now, except I'm using the first one, and that by elimination, I'll go with the rest. Viking has also agreed it's D. Yeah. Then more to talk about the, I think we've done this one we did previously. So various you've done this paper before, but people don't seem to remember the answers or the discussions. So should we change the paper?
No, please. Some of us, it's our first time.
Okay. So those who have done this before, changamkeni basi, help us. Even I don't think I was here when you discussed it. Also, that is not an intermediate in the biosynthesis of cholesterol. The biosynthesis of cholesterol. Who has the whole thing? Let us search for it then. Cholesterol biosynthesis. I think somebody can send like a picture and image of it in the group. Well. Okay, share it on the group. Then we have they have the acetyl CoA, it then goes to becomes the HMG CoA converted to mevalonate. Mevalonate is converted to IPP by the mevalonate kinase, converted to FPP and squalene. I think if you follow the flow, you will utaweza kujikumbusha on some of the things you may have gotten lost during the years you spent in school. I don't know how I can share it to the group because I don't have WhatsApp on my laptop. But basically the answer here will be the A.
Yes.
That's Trina. It has been sent to the group. So this is an example of an organized drug. What does organized mean? Organized drug. So, learning things. So unorganized drugs are crude drugs of plants, animals, origin, or having no cellular structure. They're either a mixture or chemical substance, or their decomposition products, such as resins, lattice. Okay, so give you a sentence, the difference between organized and unorganized crude drug. So organized, it's obtained from definite anatomic parts of a plant, such as flowers, leaves, fruits. And the unorganized is obtained from plants or animals by means of physical process, such as drying, incision, extract, such as. Now you see, I still don't understand from that definition. Because if I make a juice from a fruit, does that make it organized or unorganized? Okay. So organized is made up of definite tissue and cells. The other one does not have cellular structure. Oh, okay. Okay. So organized is solid in nature. The one can be solid, semi-solid, liquid in nature. Okay. Okay, so juice is it. Okay, now I get it. Yeah. Thank you for that.
Juice is unorganized.
Leaves, fruits, those are organized.
Okay, so if you take the leaves as they are, not you make a juice from the leaves.
Yeah, you crush them or you make them juice or something, you interfere with them, they become unorganized.
That's organized.
No, organized examples of organized are like rhizomes, barks, leaves, fruits, or entire plants.
So organized is you take the plant as it is. If you end up extracting it, it becomes unorganized.
Yes.
Okay.
That is, if you interfere with them, they become unorganized.
So, which is an example of an unorganized drug? Digitalis, pyrethrum, opium, clove. So, if we interfere with this, it becomes unorganized. Do we use digitalis as it is wholly?
Digitalis.
It's like you use a certain part of the
plant
that is organized. Yeah.
Yeah,
So
that's what I'm saying.
yeah.
So digitalis will be organized.
No. So for example, ama whatever the plant part, alafu tukai crush, tukai nini, whatever we are getting, maybe kama ni hiyo digitalis, hiyo ni unorganized. Like wamesema tu ni something that you have not interfered with, like the whole plant ama the fruit ama is organized. Otherwise, whatever comes out of those fruits ama ukitengeneza the leaves, the barks, whatever you do with them and you get an extract from them, it becomes unorganized. For example, like dried latex, like dried juice, dried extract.
But you can use the dried leaves of the digitalis. Okay. So as you said, when you use it as crude, like as wholly, if whether it's a stem or whatever it is, if you use it as whole, that is organized. But if you have to carry out some extraction or make a juice or make wax or make stuff from it, then becomes unorganized. Isn't it?
Yeah, from my understanding, yeah. Now the chincona bark is organized, and the digitalis inakuwa unorganized.
But chincona, you have to extract the quinine. I'm not going to take it as
the chincona bark organized.
Okay.
Yeah. And the digitalis you get from the chincona bark becomes unorganized.
I think
let's go to the other choices. Pyrethrum. Pyrethrum, we use it as whole as it is. We can all agree it's organized, right? When you're using insecticides, it's, oh my lord, so it's, hey. Okay, so opium, is it organized or unorganized? Who are you?
It's unorganized.
It's unorganized. Okay. Clove, that's organized, right? Yeah. That's definitely. So digitalis, you're saying it's also unorganized.
Yes.
There's an image I've sent a group.
And an example of
Digitalis is
unorganized drug
organized.
is opium.
From the group, the digitalis is organized and opium is unorganized.
Digitalis is organized.
Yes, from
From the definition they gave, they said the drug obtained from the direct part of the plant and containing cellular, maybe that one doesn't contain cellular tissues, I don't know. And containing cellular tissues, they are called as organized. Obtained from the plant, directly from the plant, and they are containing cellular tissues. That is, for example, like leaves and the barks and the fruits.
We can't have to answer. Based on the one that has been put in the group, it's saying digitally is collectors using center organized. Just choose the list. People can't choose the list. And the opium, papain, aloe, and kino. Aloe is unorganized. Okay. Okay. I would have thought it's organized. But aloe is unorganized, apparently. So, but we have understood the differences. I think the challenge would be now identifying base of the plants. Like tumeshika definition, what are the differences between organized and unorganized. But now when you start thinking it through, you have to know how the plants are actually used or obtained, how the name is obtained. I think that is where taswisi inaingilia. So do we have any more grievances on number 74? Somebody said opium is an extract of the poppy plant. All the other are mentioning plant names, not extracts. Oh, okay. Okay. So all the following are non-absorbable sutures except surgical catgut, silk sutures, cotton sutures, metallic sutures. Hey, doctors, tell us.
surgical catgut.
So surgical catgut is absorbable.
It's
Yes.
mainly used in CS, and nowadays you don't go to be removed; they are absorbed by the body. So
Okay, that's nice. Higher. The process of clonal propagation involves all the following except culture initiation, culture inhibition, subculture for shoot multiplication, rooting of plantlets. And thought I knew the answer until I've seen something new. So clonal propagation involves all the following except on earth. Sana sent a few more switches for the more examples of absorbable and non-absorbable sutures. So for the propagation, initiation, multiplication. Has somebody found them? No, no. Once we have stage 1, we have the initiation of aseptic cultures. Stage 2, multiplication. Stage 3, rooting of in vitro formed shoots. And stage 4 is a transplantation. So culture initiation is there. Rooting from subculture of shoot multiplication is there. Rooting of the plantlet is there. So it's a culture inhibition that's not there. So 76 is B. The bacterial species most commonly used in transformed root culture is our botanist. Come through. Fine.
Agrobacterium tumefaciens.
Here we go. I don't know what all the rest do. At least I know Agrobacterium tumefaciens used to transform root cultures. Okay. Human insulin is produced by recombinant DNA modification of
E. coli.
nice.
Can we call it a day here, please?
No. Yes. No.
Not yet.
Thank you, everyone.
You've been told not yet. You know, you're the only one tired. But yeah, anyway.
No, I see even others writing on the group. People have just said that and people have left the
Oh, okay. No, it's okay. I'm cool.
Thank you.